Hello and welcome back to the Biochemistry for Health Sciences channel. In this video, we shall discuss the intrinsic pathway of apoptosis. Cell death by apoptosis is carried out by activated executioner caspases such as caspase 3, 6, and 7. And we'll talk about these executioner caspases and what they do in a future video. The activation of these executioner caspases either occurs through the intrinsic pathway, where the signal comes from within the cell, or through the extrinsic pathway, where the signal comes from outside the cell. In this video, we shall look at how the intrinsic pathway causes the activation of executioner caspases 3, 6, and 7, which later on in a future video we'll see how these caspases go on to carry out apoptosis. Before we discuss the intrinsic apoptosis pathway, a few words about the beta cell lymphoma 2 or the BCL2 family of proteins, which contain a domain called the BH domain or the BCL2 homology domain. This family of proteins can be divided into those that promote apoptosis or pro-apoptotic, as well as those that suppress apoptosis or anti-apoptotic. The pro-apoptotic include the dead effectors or the Bex family, as well as the BH3 only family, while the anti-apoptotic proteins include the BCL2 family. Examples of proteins that belong to each category is given in this list. So here you see examples of the Bex family, here you see examples of the BH3 only family, and here you see examples of the anti-apoptotic BCL2 family. Note that all of these proteins have BH domain. So the BH3 only family has only one BH domain, that is a BH3 domain, and that's why we call this the BH3 only family, while the rest have several domains. This one here, the Bex family, have BH3, BH1, BH2, the BCL2 family have BH4, BH3, BH1, BH2, and some proteins also have a TM or transmembrane domain that allows them to embed in membranes such as the outer mitochondrial membrane. So that's what this TM here stands for, transmembrane domain. Now, why are these domains important, the BH domains? Because these proteins interact with one another through their BH domains. So you can see these proteins interact with one another through their BH domains. So what do these BCL proteins do in the intrinsic pathway? The dead effectors, they actually form pores in the outer mitochondrial membrane. So the outer mitochondrial membrane is here. This is the inner membrane. So these proteins actually assemble together to form pores. And these pores this pore formation leads to the membrane, mitochondrial outer membrane permeabilization, or MOMP, MOMP, which stands for mitochondrial outer membrane permeabilization. So basically the outer membrane becomes permeable, and we'll see what that leads to. But eventually this will cause intrinsic apoptosis, leading to cell death. The anti-apoptotic BCL2 proteins, these proteins inhibit the dead effectors. So these proteins inhibit the dead effectors. And if the dead effector proteins are inhibited, then there won't be any pore formation, there will not be any MOMP. The intrinsic pathway does not happen, and the cell does not die. Instead, the cell survives. So in other words, these anti-apoptotic BCL2 proteins, they promote cell survival, not apoptosis. You can see that some of these proteins, BCL2 proteins, are very stable. Their stability, their half-lives are in hours, while some of them, like MCL1, their half-life is in minutes, very unstable. A1 is in minutes. They very quickly become ubiquinated, and then they undergo degradation by the proteasome. So cells that are dependent on, say, these proteins, MCL1 and A1, these cells will die if their RNA protein synthesis is impaired. And that's because these proteins are very unstable, so they would not be able to prevent apoptosis or promote cell survival. So if anything goes wrong with RNA protein synthesis and your cell depends on these two anti-apoptotic proteins, then the cell has a very low chance of survival. And then we have the pro-apoptotic BH3 only proteins. So these are pro-apoptotic. So what they do, they will bind and neutralize the anti-apoptotic proteins. So these proteins will bind and neutralize the anti-apoptotic proteins. The BH3 only proteins can also directly bind and activate dead effectors. So they can directly activate dead effectors. So both of these mechanisms, either by inhibiting this or by activating this is going to lead to MOMP. That's going to cause intrinsic pathway to be triggered, and that's going to result in cell death by apoptosis. Two more proteins that is good to know before we talk about apoptosis. One is cytochrome C that we have discussed in the electron transport chain. This is found in the inner mitochondrial membrane. And the second is called the second mitochondria-derived activator of caspase, or SMAC. You can see that in the space between the inner and the outer mitochondrial membrane. This protein is also called Diablo, which stands for direct IAP binding protein with low PI. So keep these two proteins in mind, cytochrome C as well as SMAC or Diablo. We'll come across this later in this video. Okay, with that background on these proteins, I think we are in the position now to understand this intrinsic apoptosis story. So let's look at this process where intrinsic apoptosis will go and activate the executioner caspases. First, let's look at a healthy, a non-stressed cell. So in a healthy cell, there are survival signal chemicals that are coming to the healthy cell. For example, they signal human epidermal growth factor. These signal will bind their respective receptors. So, for example, epidermal growth factor will bind EGFR, epidermal growth factor receptor. And when the signal binds the receptor, this will trigger a signaling pathway. And what this pathway, the end result of this pathway is the production of anti-apoptotic BCL2 proteins. So in a healthy cell, we see a lot of these anti-apoptotic BCL2 proteins that may mainly reside, they will mainly go to the outer mitochondrial membrane. And in the outer mitochondrial membrane, what they do is they will bind with the dead effectors. They will bind with the dead effectors. They will inactivate the dead effectors. So basically, these dead effectors cannot kill the mitochondria, cannot form pores in the mitochondria, and therefore the cell survives. And that's what we see in the healthy state. So this binding is mostly on Bex and Beck, while BOC is regulated by the ERAD, the endoplasmic reticulum-associated degradation pathway. Now, what about a cell that is stressed? So, in a stressed cell that is facing some sort of stress, the stress is going to activate various transcription factors. So, for example, stress with the DNA is going to activate p53. If there is a growth factor deprivation, that is the stress, that will activate FOXO13. If there is ER stress, that will activate CHOP. And then these transcription factors will go on to increase the expression of pro-apoptotic BH3 only proteins. For example, p53 will induce the transcription of these BH3 only proteins such as BEX, NOXA, as well as PUMA. So basically, stress will increase BH3 only proteins in the cell. These BH3 only proteins, they may reside in the cytoplasm, but during the stress time, they will travel to the mitochondria. And we know that survival factors, if a cell receives survival factors, that can inhibit the translocation of these BH3 proteins from the cytoplasm to the mitochondria. So once these proteins go to the mitochondria, then some of these BH3 only proteins such as BIM, PUMA, as well as TBIT, which is basically a cleaved part of BIT, these will bind and inhibit all anti-apoptotic BCL2 proteins. So some of these proteins are very potent. They will inhibit all anti-apoptotic proteins. Other BH3 only proteins are less potent. For example, NOXA, BAD, BIC, and HRK. These are less potent. They bind and inhibit only certain anti-apoptotic proteins. And as mentioned earlier, some of these BH3 only proteins, they can directly activate the dead effectors such as BEX and BAD to trigger apoptosis, even in the absence of anti-apoptotic proteins. So what's the outcome, whether these BH3 only proteins inhibit the anti-apoptotic proteins or directly activate the dead effectors? The outcome is that these dead effectors are now free. Okay, they are free. They are no more combined with or neutralized by the anti-apoptotic proteins. So they are now free to do whatever they have to do. Now, it has been observed that the BH3 only proteins that are involved in apoptosis depends on the type of stress as well as the type of cell. So, for example, the BH3 only protein BIM is important for elimination of self-reactive beta B and T cells and removal of excess B and T cells after immune response. The BH3 only proteins BIM and PUMA, they're important for apoptosis due to growth factor deprivation, ER stress, changes in the calcium level, as well as in response to glucocorticoids. P53 promotes apoptosis by increasing these two BH3 only proteins, PUMA and NOXA. BID is activated by caspase 8. We'll see that later. The extrinsic pathway through the FAS that it will activate BID. And BID can also be activated directly by granzyme. These are proteins that come from cytotoxic T cells or natural killer cells. So now we have the dead effectors that are free, and now these dead effectors such as BEX and BEC, they can assemble or oligomerize with one another to form pores or channels in the outer mitochondrial membrane. So you can see here they are forming pores or channels in the outer mitochondrial membrane, and that's going to cause MOMP, mitochondrial outer membrane permeabilization. And now that the outer membrane is permeable, it allows a lot of chemicals to leak out of the mitochondria. For example, calcium, protons, several mitochondrial proteins will leak out from the mitochondria into the cytoplasm. And two of these proteins that is important to note that leak out include cytochrome C and SMEC or Diablo. Now, the mechanisms that regulate MOMP and cytochrome C release is still uncertain, but some reports suggest that there might be a protein involved called mitochondrial voltage-dependent anion channel, or VDAC. This is a transport protein that allows things to travel between the mitochondria and the cytoplasm. Now, in the next step, the cytochrome C that has been released due to MOMP, as well as ATP, will bind another protein called apoptotic protease activating factor 1, or APAF1. So this binding of cytochrome C to APAF1 will change the shape of APAF1, the conformation of this protein changes, and that results in the exposure of a domain in this protein called caspase activation and recruitment domain, or CARD. And we talked about this CARD protein in a previous video on caspases. So if you look at the APAF protein, you can see it has a CARD domain that now becomes exposed. So this leads to APAF1 oligomerization via CARD-CARD interaction. So one CARD of one APAF1 interacts with a CARD of another APAF1, and this leads to the formation of a heptameric wheel-like structure called the apoptosome. So now we see this structure that you can see made up of APAF as well as cytochrome C called the apoptosome. Now in the next step, the zymogen, the inactive pro-caspase 9, this protein also has a CARD domain. Okay, a CARD domain is also found in this pro-caspase 9. So what's going to happen is the CARD of the APAF, this CARD in the APAF, is going to interact with the CARD of pro-caspase 9, and that is going to bring this pro-caspase 9 towards the APAF. So once this several pro-caspase 9 comes to the apoptosome, the proximity of this pro-caspase 9 to one another will force it to form dimers, and then it will cleave itself to form active caspase 9. So the pro-caspase 9, the zymogen, will be converted into active caspase 9. And then the active caspase 9 is now going to cleave and activate the executioner caspases such as 3,7, and later in a future video we'll see how these executioner caspases go on to cleave various protein targets in the cell that eventually causes apoptosis. Okay, so that will be something we'll talk about in a future video. The role of executioner caspases, what do they do to cause apoptosis of the cell? So this is basically how the intrinsic apoptosis pathway ends up activating the executioner caspases. A few words about these proteins called inhibitor of apoptosis or IAP family of proteins. Now these IAPs, they have a domain called BIR, which is baculoviral IAP repeat domain or BIR domain. And they use this BIR domain to bind and inhibit the activity of various caspases. And by inhibiting the activity of caspases, they can inhibit apoptosis. Examples of IAPs include XIAP, X chromosome link IAP, CIAP12, NAIP, surviving, living, and so on. So XIAP, for example, blocks the activity of caspase 9 as well as the executioner caspases 3, 7. IAP expression in research is actually used as an indicator of enhanced cell survival. So if you see a lot of IAPs in the cell, that means the cell is in a survival mode. So as we mentioned earlier, so besides cytochrome C, another protein that leaks out of the mitochondria is the SMEC or diablo. So these SMECs or diablos, what they do is they will bind and inhibit IAPs. Okay, so they will bind and inhibit the IAPs, and thereby by inhibiting IAPs, the IAPs cannot block the caspases. So if the caspases are not blocked anymore, that is going to lead to apoptosis. Before we end this video, a few words about the medical aspects of intrinsic apoptosis. Number one, excess anti-apoptotic or pro-survival protein, of course, is bad, because remember this anti-apoptotic proteins or pro-survival proteins, they block apoptosis, right? They block apoptosis. So if we have excess of these proteins, that's going to prevent the death of excess or self-reactive beta or T lymphocytes, and that is going to cause or increase the chances of getting autoimmune diseases. So if there's a lot of these proteins here, then it's going to increase the chances of autoimmune disease. And that's also going to prevent the death of cancer cells. Okay, it's going to prevent apoptosis in cancer cells. So that's going to result in cancer formation. And it's also going to cause cancer cells to become more resistant to anti-cancer drugs. Okay, so excess of these proteins is bad for autoimmune diseases as well as for cancer. Now, on the other hand, the BH3 only proteins promote apoptosis. So there's drugs such as venetoclax. This is a small molecule BH3 mimetic. This drug was developed for cancers that depend on BCL2 production for their survival, such as chronic lymphocyte leukemia, CLL, as well as acute myeloid leukemia or AML. So these type of cancers, they produce a lot of BCL2 anti-apoptotic proteins, and that prevents them from undergoing apoptosis, and that's why they survive. So by giving this drug, this BH3 only mimetic drug, it is going to go and inhibit the BCL2. And if it inhibits the BCL2, now these cancer cells are going to go through apoptosis and die. Now, other drugs were also developed. So besides targeting BCL2, there were drugs that were designed to target BCL-XL as well as MCL1. However, they do have toxicity issues with healthy cells. Okay, so they do have side effects with healthy cells. However, if these drugs could be joined to antibodies, and then these antibodies could specifically go to the cancer cells, then they might help destroy these cancer cells by apoptosis. So there's a lot of research going on in this area. And finally, excessive intrinsic apoptosis is linked, has been seen in degenerative diseases such as the neurodegenerative diseases, as well as ischemia reperfusion injury. So if there is too much apoptosis, that will lead to degenerative diseases. Okay, so this is basically how intrinsic apoptosis works. In the next few videos, we'll talk about extrinsic apoptosis. So until then, once again, thank you very much for supporting this channel, subscribing to this channel, and sharing this information with your friends and family. Have a wonderful day, and we'll hopefully see you in the next video.